Hi, everyone. Welcome to Brain Talks. I'm Deborah Kan, founder of Being Patient. We're going to talk today about a topic that crosses a lot of our minds. There's a lot of conflicting research out there. How much alcohol can we really drink? And is it good or is it bad for our brains? I mentioned this because there's a lot of different studies we've come across going on at the same time with a lot of different results. So we thought what we could do is start to go to some of the researchers who are delving into this topic. I'm really pleased to have with us today Dr. Feinstein, Dr. Douglas Feinstein. He is a research professor at the University of Illinois at Chicago. Thanks so much for joining us, Dr. Feinstein.
Absolutely. Glad to be able to participate. Sure.
So one of the things that I think what we should do first is tell people what aspect, what did your research look into? We don't want to give people imply that you're an alcohol researcher. You actually study neuroinflammation. So tell us a little bit about the recent study that you conducted.
So maybe I'll give a quick background about the kind of work we've been doing for the past 10 or 15 or 20 years. So neuroinflammation, which is the main topic that we're studying in the lab, is basically when you activate, types of cells in your brain. We call them glial cells. There's microglial cells. There's other types called astrocytes. These guys are normally sitting around there sort of patrolling your brain, doing useful things, keep getting rid of junk, producing trophic factors, making your neurons happy. And in a variety of diseases like Alzheimer's, but also MS, Parkinson's, even aging, the cells get activated in ways that can become, toxic or detrimental. And so we've been studying that. And that sort of led up, that's my expertise. And then about 2 years ago, I got involved in an alcohol project, which was to look at the effects of alcohol on these cells in the brains of animal models and also in cell culture. So that's the background for the whole thing. And also I have a lot of the work we've done over the years here in my lab has focused on Alzheimer's disease and what leads to plaque accumulation, but never previously looked at alcohol. So I've just put these two things together for the first time, and luckily we have some interesting results. So that's what.
So what, I guess, this is the drum roll moment. What is it that you found? And we should say, like you said, this is an animal study. It's not a human trial. So presumably it's a mouse study. Is that right?
Correct. Yeah, it's a mouse study. Well, it's a rat study. And to be totally clear, we actually haven't even done the studies yet in the animals. We're isolating cells from the brains of animals and then treating them in culture with alcohol. And I guess the gist of what we found, which was really surprising, is that when we treat the cells, and we're using microglial cells, again, a specific cell type, and we treat them with alcohol, and it's a very high dose, so many limitations. When we look at the cells the next day and for their ability to remove beta amyloid from the cell dish, it was significantly impaired. Roughly 15%, and we only measured that over a course of an hour, so who's to say what would have happened later on. But that's the major effect, giving pretty high dose of alcohol to a bunch of happy cells. They don't like to take up and remove beta amyloid as well as the control cells.
And just to put context around this, the microglia are often referred to as the janitors in our brain. They sweep through and clean out all the gunk, so to speak, making it really difficult for plaque to build up in our brains. Is that a good layman's explanation?
That's a great explanation, totally. So you mentioned at the very beginning a lot of controversy. So controversy is the fact that when we talk about neuroinflammation, or neurodegeneration, these cells get activated and produce toxic molecules. But you have to remember that cells are not normally there to produce toxic molecules. These guys are there to remove, like you said, remove debris and produce trophic factors. And what we sort of found is that the alcohol doesn't necessarily cause them to make more toxic factors, but reduces their ability to carry out their beneficial functions. So that's also sort of like a yin and yang of this process. So yeah, they're normally there to one of their beneficial features is to remove amyloid as well as other junk by a whole variety of processes. And we happen to focus on one of them, but there's several others which we hope to get to in the future as well.
So we had a question come in earlier about when someone obviously delved into this study and was wondering why so much alcohol? Like, why did you douse? Like, is that a realistic explanation if you're kind of drowning these cells in alcohol to see what the result will be?
Yeah, that's a great question, and so many answers to that. I've been thinking about it. There was actually a comment on one of the reviewers' comments on the paper when we got it back for revisions. So first, oftentimes carrying out initial studies, we want to treat the cells with high doses of stuff to see if anything's going to happen. If you hit them with a very high dose and nothing happens, then maybe you don't want to go back and use lower doses. We make sure that the high dose doesn't kill the cells, which it didn't. So that was one of the reasons. A second thing, in terms of actual human consumption, the equivalent of what we used is pretty high. It's about 0.3%, and I'm sure everyone knows that, you know, the drinking limit, the legal limits in a lot of states is 0.08%. So we're about 3 to 4 times higher. Okay, and you know, the numbers are that I find is this is the equivalent of drinking 6 or 8 beers or shots or something like that within an hour, or which is the equivalent of about a bottle of wine in an hour. And this is a lot.
That's a lot.
It's a lot. Hopefully, hopefully it's not for most people, but it's not for many people. So for many people, that's normal, and you know, or even higher levels. And if your alcohol blood levels are that high, you're not in a good shape. You're unless you're used to it, you may be in a stupor, maybe unconscious. If you're much higher than 0.3%, I think 0.4% just could be lethal. You know, it depends on your health, your age, your gender, your diet, and all those types of things. But it's a high amount. Now, but another thing addressing the same question is what we do is we add the alcohol to the cells at a very high concentration up front, and then they're in a chamber because they need, you know, 37 degrees, and they need carbon dioxide and oxygen to survive. And during that time, the alcohol is evaporating. Now, so to be honest, we did not measure the levels during the protocol, which was 24 hours. I suspect that by the end of the protocol, it's probably pretty low. So this would be like, I would say the average is probably, who knows, midway between those two. And this is actually very physiologically relevant because in humans, you drink a bit, even if you're drinking normal amounts, and it's being removed from your gut, it's being evaporated. So actually, to some extent, it's a little bit more physiologically relevant, but it's still a lot.
Yeah, we have another question. I'm hearing it on your side.
Turn it down.
Yeah, sorry. Another question about, this is a good point. Do animals react to alcohol in the same way that humans do? So if you're giving, I mean, I know it hasn't been, you haven't tested it actually on the animal, but how relevant is this test on animals versus humans?
So this is the crux of basic research, which, you know, can be asked of any scientific studies. So I think, you know, it's a good model, and from other studies that are being carried out that I'm aware of in alcohol research, and there's actually, I mentioned earlier, well, we were talking that the work I'm doing is part of a larger program project at UIC, and there's 5 or 6 other investigators who are looking at alcohol effects in animal models. And from their results, which involve consumption and overconsumption and also withdrawal, many of the behavioral effects that they see replicate fairly well what happens in humans. The withdrawal effects, the addictive effects. So in that aspect, I think it is pretty similar. But there may be 1 or 2 papers that have been published looking at the effects of alcohol consumption in Alzheimer's models, mouse models or rat models. So we really don't know yet. It has to be done.
And there is a lot that is unknown. You know, we always hear like in diets like the Mind diet say, yeah, a glass of red wine is good for you. And we hear that a lot. It's like a glass of wine, a red wine a night is actually beneficial to your health, to your brain.
Yeah.
But I've always, when you dig deeper into that, I've always heard of it spoken in the context of an element that comes from the deep red color of a grape, right? So I think it's called resveratrol, which is, yeah, did I mispronounce it? What is it called?
Yeah, that's right.
Yeah, resveratrol. And resveratrol is something we know that helps open the blood vessels, right? And therefore allows better blood flow even to our brain. So how much can we count on that to say, well, yeah, a glass of red wine a night is actually it's not bad for you, or is it?
Yeah. So great. Yeah. So first off, reading about wines. So white wines are also made from the same red grapes, right? They're just taken off the fermentation process a bit earlier, so they don't pull out the pigments. But they still pull out a lot of the same chemicals. I'm not sure how much resveratrol is in white wine, but there's a lot more in red wine. But they're different in a lot of other factors. Reading the literature and reading some of the sort of meta reviews where somebody has reviewed a lot of the reports, I'm pretty confident that taking low amounts, a glass of wine a day, as recommended by or suggested by the Mind diet and other diets, is probably going to be good for you if you've been doing it. And that said, if you're not drinking anything, to consider to start drinking any alcohol at all as a means to reduce the risk of AD or reduce development of disease, I think is not a good idea. Clearly, your body is not used to it. But if you've been doing that all your life, well, for many years, that's probably fine. I mean, I would, the glass, I think the recommended is 1 to 2 glasses per day. But it's very conflicting. There are reports that even moderate drinking can have deleterious effects. And it's a controversial area. So
yeah, I guess there's still a lot we don't know. But tell us a little bit about the broader research, some of the results that they're seeing at the University of Illinois, Chicago. What are some of the things that you're learning, maybe not in context to your particular study, but overall, if this is a large scale study on the impact of alcohol, are there any preliminary takeaways?
Right, sure. So the larger project here, which I should mention, there's a PI for the entire grant. His name is Subhash Pandey, Department of Psychiatry at UIC. And the overall goal of that project is looking at an area of genetics. And I should say I'm really a molecular biologist by training. So we're looking at an area of genetics called epigenetics. And what this focuses on is the fact that your environment, your diet, your social interactions, everything affects your DNA, not changing the bases, not to cause mutations, but it's to cause epigenetic changes, so outside of the genes, which means that it's modifying the structure of the chromatin, it's modifying the bases, but not changing them. And the thing is, these changes are inheritable. That when your cells divide, the new cells can have the same changes. When you have children, your children can inherit those. So it's looking at the epigenetic effects of alcohol in different parts of the brain. And in particular, one big aspect is during withdrawal. It seems that withdrawal causes a lot of epigenetic changes, which contribute to the adverse effects, depression, anxiety, addiction. And the benefit is that there are drugs now that can be used that are FDA approved to prevent that from happening. So that's the overall goal. How does alcohol cause epigenetic changes? And if we treat the animals or cells or people eventually with inhibitors, can we prevent that? So in the context of Alzheimer's disease, what we haven't published, but what we're also working on is, we saw these changes in these genes which affect the clearance of beta amyloid. So how does that happen? So the idea is that we're causing these epigenetic changes. And that's like one of the next stages. How is this happening? Now, we know alcohol is doing it, but how is it doing it? So that's kind of the broad context of this whole program project.
Is it possible that our genetic like the way that our genes react to things are different? It's different for everyone. We all have our unique genetic code. Like, so for example, you take a disease like cancer and you talk about people, some people can smoke all their lives and never get cancer, and other people will get it, in a matter of years. Should this study is like perhaps people's microglia will, the whole system will act differently depending on your own genetic code?
Yeah, I think that's absolutely true. I think, again, when you ask why are we using a high dose, one of the reasons is to sort of overwhelm the system and sort of get everything going in the right direction. If we use moderate doses or low doses, there are so many differences between people. I mean, there's millions. Well, I think we do a lot of genetics here. We're comparing, right now we're comparing identical twins, and they have a few differences, but just like siblings have on the average 50% differences. And so we're all different by hundreds or thousands and millions of genes. Any process you look at in your body, including removing beta amyloid, involves dozens or hundreds of genes. So yeah, everyone's got many differences, and the effects person by person are going to be very different, which is why it takes so many people to do clinical trials. Everyone, between that and your diet and all the other factors, it makes things really difficult to get reproducible results. Yeah.
I thought also what you said about withdrawal is quite interesting. We had a question about binge drinking versus just like one or two a night, and really like, is that worse? I mean, if we're talking about like the way that you're describing it to me, it makes me feel like, or maybe I'm misinterpreting it, that it's actually that withdrawal period that can cause damage and lead to other things like anxiety or depression, which we know are not good for our brains.
Right.
So are there any studies about different types of drinking habits, like binge drinking versus one or two a night? Because I think if you're abusive, with alcohol, that's definitely bad for your health, and I don't think anyone would argue that. But are we looking at these studies as someone like me, 1 to 2 glasses a night? It relaxes me, and I'm not drunk, but I like it.
Exactly right.
So there are different categories in there.
I think, again, you're absolutely right. Now, so as you know, I'm not an alcohol physiologist, so I'm sure other folks in this program project know much better than I. My gut feeling is that, yeah, binge drinking is certainly not good for you, and the responses are going to be different. But I think the question of withdrawal, that is an amazingly important question, because there's withdrawal from maybe the physiology that's involved in withdrawal from, I don't even know if withdrawal can't be used when you're taking a single drink or 2 drinks. I don't think withdrawal applies to that situation. Withdrawal applies to the situations where someone has already established a strong drinking habit. Not want to say addictive, but some type of strong amount of drinking, which then is cut, and that might be more physiologically robust response. So as someone who's just gotten into the alcohol field 2 years ago, this is my initial takeaways. But these are going to be, you're basically making me think about a lot of really interesting studies that we should start thinking to do.
Do we even know? Do we even know why alcohol is addictive?
Well, I mean, so many people probably know that the neurotransmitter dopamine plays a large role in addictive behavior, and there's data which shows that alcohol increases the release of dopamine in certain parts of your brain. So that's one of the reasons why it's addictive. And withdrawal, to some extent, again, as a new scientist in this area, is an effort from your brain to sort of compensate for the lack of dopamine in other ways. And then when you then follow it up with another drink, you're taking that on top of a compensatory reaction, and so it's a circular process, which it's ongoing. So it's a self-replicating circle. It's not a good thing. But yeah, it's a large extent dopamine-related addiction. Yes.
So if according to your
let me step back, which is very interesting, because there's a really interesting connection between the role of dopamine and addiction and alcohol abuse and Alzheimer's disease. And one of there are a lot of neurotransmitters which are dysregulated in your brain if you have Alzheimer's or MS or PD. We study one of them, which is called noradrenaline, which is highly related to dopamine. Noradrenaline also plays a role in alcohol addiction and anxiety. So there may be some really interesting similarities or common pathways that alcohol affects and the process of Alzheimer's disease affects. And it's potentially that the two together are synergistic. I mean, we don't know yet.
So one thing that, and we talked about this at the beginning, there are so many contradictions. People are confused because we really don't know, right? So as a scientist, how far away do you believe you are, or science is, in determining some of these answers? I mean, in relation to Alzheimer's disease, how far away are we to understanding whether or not, a glass or two? We got another comment from a viewer saying, I take my mom camping or to a place, and I give her a glass or two of wine, and it really helps her calm down. Yeah. So how far are we away from getting some of these answers?
Oh, a while. I mean, in a practical sense, what I'm hoping to do is get funding to carry out the comparable studies and some animal models. And if we get funding to do that, it'll be 2 years. And I'm hoping that other researchers will pick up on what we've just published and start looking at it themselves. If that happens, then things may accelerate a bit, and maybe these two areas will advance a bit quicker. We've been looking for drugs for AD for a long time, and there's nothing out there yet, which is really, has very high promises. This is a subtle part of the disease process. So, maybe it'll be sooner. Maybe about 10 years till we can really say, you are okay to have a glass of red wine every night, but based on your DNA or your background, you really shouldn't. So maybe 10 years.
Right. And so if we were to look at your study down the pipeline, what you're hoping to find out is if in fact you don't drink, will it actually help you prevent Alzheimer's disease by keeping the system in place as it was designed, the microglia, the janitors of our brain, working in the way that they were designed to work. Is that the right way to put it?
That's a good way to put it. Well, so not drinking will keep them in line to what they're doing. I think there's a practicality that a lot of people, for whatever reasons, do want to have a glass of wine. And if it's the case that this does cause some dysregulation, then maybe we could think about ways to allow that to continue, but to provide, a medication, even something as simple as ibuprofen, not that ibuprofen is the answer, but something, or aspirin, which might prevent alcohol from having those effects on the microglial cells. I don't think we can prevent people from their habits. And so something that we have to live with and maybe adjust to. That's, I don't know if it's pessimistic or optimistic, but it's a practical solution.
I think that's a good way to look at it. And I think we can wrap it up there to say that, there's still a lot more research. I think it really helps to talk to researchers such as yourself to understand the perspective and some of your preliminary findings that certainly are valid to explore further, to give us, people impacted by the disease, people worried about brain health, more information on making that decision personally a glass or two, stop drinking, what have you. So thank you so much for joining us and for your research. I think people will find it really informative. This is not a topic we're going to have fast, easy answers to. Like even myself, I would love you to say to me, oh, just have a glass or two. But with that, but with that said, we definitely will stay with this topic and look at the different aspects of research and the findings around it as they pertain to brain health. So
Great.
Dr. Feinstein, thank you so much for joining us for this talk. I found it very interesting and helpful.
Good. Well, thank you for letting me talk to a few people, and hope I've gotten some information across, and I'm always here. I'm more than happy to answer any questions that anyone has. So please send them this way. Thank you for what you're doing. It's great.
All right. Thanks so much.
What do I do?